Thank you for joining us. The topic of this presentation will be low voltage electron microscopy for materials and life science applications. I will start with a brief introduction of the relevant electron microscopy topics, such as transmission and scanning modalities, followed by an introduction to low voltage electron microscopy. After that, we will dive into more detail about the uses and advantages of LVM for both material and life science applications. Electron microscopes are used when the objects of interest are smaller than the wavelength of visible light, which is about 400 to 700 nanometers. An electron beam has a much shorter wavelength than visible light. As the electron beam interacts with the sample, it produces a variety of different emissions. In transmission electron microscopy, or TEM, we use the transmitted electrons to form the image. TEM is excellent for high-resolution imaging, studying morphology, revealing internal structures, and performing accurate size measurements. Scanning electron microscopy, or SEM, uses a finely focused beam on the specimen, often called the electron probe. It's typically used for bulk samples where we want to examine the surface in detail. SEM is great for surface morphology, topography with large depth of field, and compositional contrast. Now let's talk about scanning transmission electron microscopy, or STEM. STEM combines the best of both worlds, the focused electron probe of a scanning electron microscope and the transmission capabilities of a traditional TEM. It's ideal for thin samples and offers outstanding high-resolution imaging. Plus, it provides excellent contrast even for light elements, making it a powerful tool for detailed structural analysis. Next, we have electron dispersive X-ray spectroscopy, or EDS. EDS is an analytical technique that can be paired with TEM, SEM, or STEM. It allows us to identify elements in a sample and even estimate their relative amounts. This makes it incredibly useful across many fields, from material science, where it's used for alloys and nanostructures, to the life sciences for applications like biological labeling or tracking nanoparticles inside cells. Now let's take a moment to look at some of the challenges that come with using high voltage TEM systems. These instruments are massive and typically require a dedicated electron microscopy facility. That means specialized infrastructure, an anti-vibration platform, a chiller or liquid nitrogen supply, and an on-site staff to keep everything running smoothly. Because these systems are usually shared within an institution, scheduling can also become a real bottleneck. And of course, both the instruments itself and the supporting infrastructure come with a significant price tag. High voltage beams can also damage sensitive samples through knock-on effects, radiolysis, or even localized heating. On top of all that, sample preparation can be extensive and time-consuming. Another important challenge with traditional high voltage TEM is enhancing image contrast. Historically, this has relied on staining, most commonly using a combination of uranyl acetate and lead citrate. But these chemicals come with serious drawbacks. Uranyl acetate is not only radioactive, but also carcinogenic, and lead citrate, likewise, is a known carcinogen. So while staining has been a standard approach for decades, it introduces significant health, safety, and regulatory concerns. Given all these limitations, it's clear why a different approach is needed. So let's turn to the LVEM system and what it brings to the table. Let's move to the next section, an introduction to low voltage electron microscopy. There are two main reasons why low voltage electron microscopy, or LVEM, is so valuable. First, operating at low voltage dramatically increases image contrast for low atomic number materials, things like biological samples, polymers, and other light element structures that are traditionally difficult to visualize. Second, using low voltage allows us to replace bulky electromagnetic lenses with compact permanent magnet lenses that don't require any cooling. And that's really the key to why the entire LVM column can be miniaturized, making the system far more accessible and easier to integrate into standard lab environments. Let's take a closer look at how accelerating voltage affects electron scattering. At low voltage, around 5 kilovolts, electrons scatter much more strongly from the atoms. This produces higher image contrast, especially for light elements like carbon, hydrogen, and oxygen. At higher voltage, say 100 kilovolts, electrons scatter less and mostly in the forward direction. This means they penetrate deeper and contrast for light elements is reduced. So in short, low voltage gives you more contrast for delicate, thin, low Z samples, and high voltage gives you more penetration and resolution for thicker or heavy element materials. Now let's talk about contrast and beam damage, two key factors in electron microscopy. As we just saw, contrast depends on how strongly electrons scatter in your sample. Low voltage electrons scatter more, which boosts contrast for light elements and delicate materials. But every electron that interacts with the sample also has the potential to cause damage. High voltage electrons can knock atoms out of position, heat the sample, or break chemical bonds. Low voltage electrons reduce damage, such as knock-on damage, though inelastic effects like radiolysis can still occur. The goal is always to balance image quality with preserving the sample, getting the highest contrast while minimizing damage. One of the great advantages of LVEM is how simple sample preparation can be. In many cases, post-staining can be completely eliminated. There's no real need for heavy metal stains. Urinal acetate is not mandatory, and if staining is needed, urinal-free alternatives can often provide similar or even better contrast. That means the process is far safer, less toxic, and completely free of heavy metal and radioactive materials. Because low voltage electrons scatter more strongly, even subtle differences in density within the sample become easy to detect. This means you can see fine structural details with fewer artifacts than you would get with conventional heavy metal staining. Let's look at a direct comparison of image contrast. When we measure signal to noise ratio, or SNR, LVM provides roughly 10 times higher SNR than traditional high voltage TEM. This means LVM images are much clearer, making it easier to see fine details in delicate or low density samples. In short, higher contrast and clearer images translate to faster, more reliable analysis without the need for extensive staining. One of the biggest advantages of LVM is that it can be installed directly in your own lab. This gives you routine, on demand access, no need to schedule time in a shared core EM facility. The workflow is simple and fast. Prepare your sample and move straight to imaging, getting results quickly and efficiently. There's no need to book a core facility or transport your sample elsewhere. Here are some examples of in-lab low-voltage electron microscopes. The LVM5 and LVM25E from DeLong Instruments are benchtop and compact systems designed for routine use directly in your lab. They combine high-contrast imaging with simple sample preparation and quick turnaround, making advanced electron microscopy accessible without the need for a shared facility. Let me introduce the LVM5 benchtop electron microscope. First introduced in 2003, the LVM5 is an entry-level system designed to bring high-quality electron microscopy directly into your lab. It offers multiple imaging modes at 5 kV, transmission mode with 1.2 nanometer resolution, scanning mode with 4 nanometer resolution, and STEM mode with 2.5 nanometer resolution. The LVM5 is most commonly used for nanomaterials analysis and general in-lab electron microscopy, providing a compact, easy-to-use solution for routine imaging. The LVM5 is designed with a benchtop form factor, making it ideal for standard laboratory spaces. Next, let's look at the LVM25E Compact Electron Microscope. Introduced in 2022, the LVM25E is the most advanced low-voltage electron microscope in the lineup. It offers multiple imaging modes, transmission mode at 25 kV with as low as 1 nanometer resolution, STEM at 10 and 15 kV and resolutions as good as 1.0 nanometers, SEM at 15 kV with 4 nanometer resolution, and it also includes EDS for elemental analysis. The LVM25E is ideally suited for biological thin sections, virus imaging, and general in-lab electron microscopy, combining high-resolution imaging with compact, lab-friendly operation. The LVM25E is designed with a compact, space-efficient form factor, making it suitable for standard laboratory environments. No special facilities are required for installation. This means you can bring electron microscopy directly into your lab without needing a dedicated EM facility or extensive infrastructure. Let's look at some of the key technical features of LVM systems, focusing on control and software. These microscopes are optimized for simplicity and ease of use. A sample exchange wizard guides you through sample loading, while intuitive physical controls let you easily adjust magnification, illumination, stage movements, and image optimization. The software interface is equally user-friendly, offering on-screen measurements, live histogram corrections, FFT analysis, and real-time imaging statistics. Together, these features make imaging straightforward, efficient, and accessible even for first-time users. Now let's focus on the unique electron optics that set LVEM apart. Both the 5 and 25 kV systems use a Schottky type field emission gun, or FEG, which delivers very high brightness and excellent spatial coherency. This allows for strong interactions between electrons in the sample, producing the high contrast images LVEM is known for. It also enables the use of permanent magnet lenses. These lenses generate no heat and require no liquid cooling, contributing to the system's compact, low maintenance design. Now let's shift gears and look at how LVEM can be applied in material science. LVM's high contrast, high resolution imaging, and in lab accessibility make it a powerful tool for analyzing nanomaterials, alloys, polymers, and other advanced materials. We'll explore specific examples and workflows that highlight its unique advantages in this field. First, let's compare some common techniques for studying morphology: AFM, DLS, and TEM. AFM, or atomic force microscopy, provides surface profiling, giving information on shape, surface roughness, and height. It offers 3D surface information, works in air or liquid, and requires minimal sample preparation. The limitations are a small scan size, lower throughput, and the potential for tip-sample interactions that distort soft materials. DLS, or dynamic light scattering, measures hydrodynamic diameter and size distribution of particles only in solution. It's simple, non-destructive, and gives average particle sizes. However, it provides no shape information, assumes spherical particles, and is sensitive to aggregates. Finally, TEM offers direct imaging, revealing particle size, shape, and internal structure with high spatial resolution, often down to the sub-nanometer level. It allows imaging of internal morphology, though height measurements require additional techniques. Here we see examples of LVM images showing nanoparticle morphology. These images highlight particle shape and size, showing how individual nanoparticles can be clearly resolved. Furthermore, we have an example of elemental analysis using EDS. By pairing EDS with STEM imaging, we can identify the metals present in a sample and even get semi-quantitative estimate of their composition. This allows us to correlate the elements' information directly with particle morphology, helping to understand material properties, including composition. For more advanced analysis, we can use software like ImageJ or ImagePro to automatically identify particles in TEM images. The software can measure particle size, shape, and distribution, and generate average sizes along with other statistical metrics. This approach streamlines data analysis, reduces manual measurements and errors, and provides robust quantitative information directly from your images. We have TEM images of different types of nanoparticles: PBS, gold, and latex, as well as carbon nanotubes. The first images show PBS nanoparticles, highlighting their uniform size and shape. The mixed sample demonstrates the morphology of these various nanomaterials. This example illustrates how LVM can capture detailed information on particle size and shape across a range of materials. Carbon is a dominant element in organic matter. In electron microscopy, imaging carbon can be challenging because it provides relatively low contrast compared to heavier elements. LVEM enhances contrast for carbon, making it possible to visualize a range of carbon-based nanomaterials, from graphene and nanotubes to polymeric nanoparticles and thin films. The capability opens up detailed studies of structure, morphology, and even subtle density variations in carbon-rich samples. Here we have TEM and SEM images of carbon nanotubes with platinum nanoparticles embedded. The carbon nanotubes form the tubular framework, while the platinum nanoparticles appear in TEM as dark or SEM as bright, highly contrasted spots distributed along the tubes. This combination highlights how LVM can resolve both light element structures like carbon and heavier elements like platinum in the same image. It's a powerful demonstration of LVM's ability to reveal detailed morphology and composition simultaneously. Here are some more images of the same material, both in dark field and bright field, again highlighting the position of the organic material in relation to the inorganic. LVEM also enables imaging of single-layer carbon materials such as graphene sheets. Even these ultra-thin structures can be clearly visualized thanks to the enhanced contrast at low voltage. In addition to direct imaging, LVEM can produce electron diffraction patterns, providing valuable information about crystallinity in single-layer graphene. This makes LVEM a powerful tool for studying the finest details of carbon-based nanomaterials. I'm excited to highlight a recent publication in MRS Advances titled Advancing Liquid Phase Electron Microscopy, Low Voltage Electron Microscopy with Graphene Liquid Cells. This work demonstrates how LVEM can be applied to study dynamic processes in liquid environments using graphene liquid cells to contain samples while maintaining high resolution imaging. It's a great example of how low voltage electron microscopy is expanding the possibilities of materials research, allowing scientists to observe nanoscale behavior in real time. Here we have TEM images of 6 nanometer cadmium selenide quantum dots. These images clearly reveal individual nanoparticles, showing uniform size and shape across the sample. Low voltage electron microscopy provides excellent contrast for these small, delicate particles, allowing precise analysis of their morphology and distribution. Shown here are TEM images of polymer vesicles. These images reveal their spherical morphology and uniform size distribution, highlighting the ability to resolve fine structural details. LVM's low voltage operation provides excellent contrast for these delicate, low density structures, making it easy to study vesicle formation, size, and internal features. This showcases the power of LVM for analyzing soft, polymer based nanomaterials. Let's now shift our focus to life science applications. LVM's ability to provide high contrast imaging of low Z materials combined with simplified sample preparation and in lab accessibility makes it an ideal tool for studying biological specimens. We'll explore examples ranging from viruses and thin tissue sections to nanoparticles within cells, showing how LVM can reveal detailed structural information without extensive staining or complex infrastructure. We're pleased to highlight our recent publication in Microscopy Today titled Low Voltage Electron Microscopy Part 1 Benchtop and Compact TEMs with Heightened Contrast for Soft Materials. This article showcases how LVM systems provide enhanced contrast for delicate, low density samples, such as biological materials, while remaining compact and accessible for routine in lab use. It's a great example of how low voltage electron microscopy is advancing practical high resolution imaging for the life sciences. LVM provides higher contrast for soft materials, making it ideal for biological applications. Here we have examples demonstrating this capability. First, DNA origami structures, where fine nanoscale features are clearly resolved. Next, gold nanoparticles inside macrophages, showing both the particles and the surrounding cellular context. Last, detailed cellular ultrastructure, highlighting membranes, organelles, and other delicate features. These images illustrate how LVM can reveal both the morphology and the internal organization of biological samples with exceptional clarity, often without the need for heavy metal staining. I'd like to highlight a recent publication titled Urinalysis Low Voltage Transmission Electron Microscopy, a powerful tool for ultrastructural studying of cyanobacterial cells. An excerpt from this paper notes that results demonstrate LVM could serve as a method to eliminate conventional, highly toxic stains such as uranyl acetate. The authors suggest that LVM not only provides high contrast ultrastructural imaging, but also opens the door to safer, more accessible workflows, making it worth further investigation for a variety of biological applications. Here we have examples of ultracellular analysis using LVEM. First, a cross-section of cilia showing detailed microtubule organization and fine structural features. Second, mouse liver tissues highlighting cellular architecture and organelle details. And last, muscle tissue, where sarcomere organization and fine ultrastructure are clearly visible. These images show how LVM provides high contrast, high resolution views of delicate biological structures, often with or without toxic heavy metal stains. I also want to highlight here a publication from the Journal of Histochemistry and Cytochemistry titled Diagnostic Electron Microscopy of Viruses with Low Voltage Electron Microscopes. An excerpt from this paper notes a remarkable advantage of low voltage TEM. Ultra-thin section samples prepared with osmium tetroxide and block contrast did not require any additional staining to image structural details. This demonstrates how LVM can simplify sample preparation while still providing high contrast and high resolution images of viral ultrastructure. Here we have examples of viral imaging using LVEM. On the left, we have an image of coronavirus. In the center, the Portugal mosquito flavivirus, and on the right, a mimivirus. These images illustrate how low voltage electron microscopy can capture viral morphology clearly, making it a powerful tool for virology research. In this slide, we have images of tobacco mosaic virus, or TMV, captured using LVEM. The filamentous structure of TMV is clearly visible, showing uniformity in size and shape along the entire particle. In this slide, we have TEM images of mouse brain tissue captured using LVEM. These images highlight nerve cross-sections, showing the arrangement of axons within the tissue. They also show myelin sheaths, clearly revealing their layered structure and organization. Here we have TEM images of kidney tissue, highlighting how LVEM can be applied in clinical nephropathology. These images reveal fine structural details of glomeruli, podocytes, and other renal components, providing critical information for diagnosing kidney diseases. LVEM's high contrast and resolution make it possible to assess delicate structures in thin sections quickly and accurately, supporting faster and more precise clinical analysis. Another publication I'd like to point out is in the Pathobiology and Emerging Techniques journal titled Clinical Validation of Low Voltage Compact Transmission Electron Microscopy for Ultrastructural Evaluation of Kidney and Heart Biopsy Samples. An expert quoted in the paper emphasizes that compact TEM is a clinically valid method for ultrastructural evaluation of renal and cardiac biopsy specimens. The study shows that compact TEM maintains a comparable ability to discriminate key diagnostic findings, demonstrating that LVEM can support accurate, routine clinical analysis while offering the advantages of a smaller, more accessible system. Shown here is a TEM image of muscle tissue captured using LVEM. The sarcomere structure is clearly visible, with individual myofibrils and filament organization easily resolved. Here we have images of the 20S proteasome captured using LVM, highlighting its capabilities for single particle analysis or pre-screening prior to cryo-TEM. Individual proteasome particles are clearly resolved, allowing detailed visualization of size, shape, and structural features. LVM provides high contrast images of these delicate protein complexes, enabling quantitative analysis and characterization. We've now seen how LVM provides high contrast, high resolution imaging across a wide range of applications, from material science and nanomaterials to virology, cellular biology, and clinical pathology. Let's take a moment to summarize the key advantages and wrap up the main takeaways from today's presentation. We honor Professor Armin DeLong, the namesake of our company. As he once said, every scientist should have the opportunity to regularly use a transmission electron microscope for his or her own research. His vision drives our mission to make high resolution electron microscopy accessible by building compact, user friendly, and affordable electron microscopes for scientists in every field. Here are just a few examples of the many users of our LVM systems around the world. Our instruments are installed globally across universities, clinical labs, research institutes, and pharmaceutical companies, supporting everything from fundamental science to diagnostic workflows and advanced material science research. This global adoption reflects the same core idea: accessible, high contrast electron microscopy that fits directly into the lab, wherever that lab may be. To summarize, low voltage TEM offers a number of key advantages that make high resolution electron microscopy more accessible than ever. First, LVEM dramatically increases the image contrast for low atomic number materials, and by using permanent magnet lenses instead of electromagnetic lenses, we're able to miniaturize the entire column, making the systems compact, stable, and easy to maintain. LVM serves as a direct replacement for traditional high-voltage TEMs, but with far lower startup costs and virtually no maintenance requirements, no service contracts, and no complex infrastructure. Installation is simple. Both the benchtop LVM5 and the compact LVM25E can be placed in small labs with minimal facility requirements, allowing electron microscopy to live where the work actually happens. And finally, LVM increases your analytical capabilities. The LVM25E offers TEM, SEM, STEM, diffraction, and EDS at 10, 15, and 25 kV. The LVM5 provides TEM, SEM, STEM, and diffraction modes at 5 kV. Together, these systems deliver high contrast, high resolution imaging in compact, benchtop, and user friendly formats, bringing electron microscopy directly into the lab. Thank you very much for your time and attention. If you have any questions or would like to discuss how LVM can support your research or clinical workflow, please reach out to us and we would be happy to advise you on all matters LVEM.